Hi and welcome everybody to the Bio+MedVis challenges at IEEE. I am one of the co-organizers of this workshop. My name is Daniel Jönsson, and the three other co-organizers, they couldn't make it here, so I forced them to record a little bit of an introduction where they talk a little bit about this challenge. First up we have Zeynep. Sorry. I should, of course, have sound. Do we have sound?
Hi, everyone. Welcome to Bio+MedVis Challenge 2022. My name is Zeynep Gümüş, and I'm one of the co-chairs of the challenge. My co-chairs are Daniel Johnson, who's there with you in person today, as well as Renate Radu in MedVis side, and Thomas Holt and myself on the BioVis side. The challenge title this year is to tame protein beasts through visualization. That is, we want to understand how we can visually best represent multiple chemical modifications on 3D protein structures and how they relate to inherited disease. Such an understanding has the potential to unlock cures for many diseases, including also complex disease. For this purpose, we have a main challenge and the redesign challenge. In the main challenge, we want to relate chemical modifications to protein 3D structure in such a way that we can visualize multiple potentially overlapping modifications in a user-intuitive way. We then want to visually relate a protein mutation and residues proximal to it that is known to be involved with a specific disease and its modification status. In the redesign challenge, we want to take an existing visualization as a starting point and then revise and improve this based on fundamental visualization principles. Thank you so much for your participation. And now my co-organizer, Tom Holt, will introduce you to the awards we have planned for the challenge winners.
Hi, Thomas here, and I have the pleasure to announce that for both of these challenges, we have a Best Challenge Award that is presented by our independent jury consisting of Bara, Haichao, and Enrico, based on the submissions and today's presentations. Both awards come with a beautiful book for the first prize. For the main challenge, that book is The Machinery of Life by David Goodsall, and for the redesign challenge, it is Immune by Philipp Detmar of Kurzgesagt. The winners will be announced and receive their certificates and prizes during the closing, so an extra reason to stay around for the whole workshop, besides the exciting program that Renata will now present.
So this brings us to today's program, which, as you can see here, is very rich. And we will start right afterwards with a talk by Enrico Massignani from the Flanders Institute of Biotechnology, who is actually one of the providers of the datasets for your challenges. And afterwards, we will have the talks by you. First, the challenge talks and then the redesign talks. And in the afternoon, we will have a short coffee break. And right after that, we will welcome our three invited speakers who have very different expertises and very different topics for their talks. First of all, we will welcome Haichao Miao from the Lawrence Livermore Institute. And then we will have Yihan Shao from the Oklahoma University, and we will conclude today's program with a talk of Barbara Kozlikova from Masaryk University. So I hope you're looking forward to these as much as I do, and sit back and enjoy.
I would also like to remind everyone that you can ask questions via Slido. We will have questions after the first talk. Between each of these challenge contributions, we will not have questions, but you're welcome to submit and talk during the break for questions there. There will also be opportunity to talk to some of the contributors, the ones that are physically here, during the poster session on Wednesday. So don't miss that. There will be a couple of ones that are displaying their posters. With that, I would like to invite Enrico up here Enrico did his PhD in Milan and is now currently doing a postdoc at the Flanders Institute for Biotechnology. And he's working with Professor Leonard Martens on protein modifications.
Okay, so good afternoon, everyone. My name is Enrico Massignani, and with this presentation, I would like to introduce the topic of this year's Bio+MedVis challenge, which is the visualization of protein post-translational modifications, or PTMs. So the challenge briefly asked you to map PTMs on protein tridimensional structures, visualize those sites where multiple PTMs could occur, and finally highlight the sites that could be mutated in rare diseases. So in the first half of this presentation, I will give you a bit of background and explain how we generated the data that you analyzed, and then I will show some examples to try and explain why some cases where visualization can be of help to researchers. So first of all, post-translational modifications consist in the covalent addition or removal of protein groups from the side chains of amino acids in proteins, and they greatly contribute to the diversity and complexity of the proteome, because from just a few thousands of unmodified proteins, they can generate potentially millions of proteoforms. And the PTMs cannot be, they're not directly encoded by the DNA, so they cannot be studied with the traditional genomics methods, but they always introduce a mass change in the proteins. So for these reasons, the main technique that is used to study PTMs is mass spectrometry. Now, in the most common MS-based proteomics workflow, proteins are extracted from proteins, then digested into peptides, and then the peptides are separated on a liquid chromatography column that is connected to a mass spectrometer. The mass spectrometer will fragment the peptides and record the mass to charge ratio of these different fragments to obtain the MSMS spectrum. These MSMS spectra are then compared to theoretical MSMS spectra to understand which peptide generated which spectra. And these theoretical MSMS spectra come from peptide search engine, which what they do essentially is that they start from a FASTA database of your reference proteome, and they simulate in silico the digestion and fragmentation steps in order to get these theoretical spectra. And they do this both for the real protein sequences and for reversed protein sequences. In this way, we obtain from the digestion of these reverse protein sequences fake peptides that can be used as negative control to assess the FDR of the results. This is a strategy that works very well for unmodified peptides, but when we introduce PTMs, we observe an exponential growth of the search space, because, of course, the search engine has to compute theoretical spectra for both unmodified and modified forms of each peptide. And the larger the search space, the more likely it is that an experimental spectrum will be matched to a theoretical spectrum at random. This means that the overall confidence score of our identification decreases, and we end up in a situation like this, where if we want to filter our results to 1% FDR, we have to throw away all the orange, the COI hits, which are the false positives, but also a good portion of the target hits. And so how we can fix this situation? Well, of course, we can search just a few PTMs, or we can improve the scoring function of the search engine. And this is where the Compomics group, where I'm doing my postdoc, comes into play, because we developed two predictors called MS2PIP and DeepLC, which can predict based on machine learning, the fragmentation spectra and retention times of peptides. And we observe that when we add this information to the features already used by the search engines, we can use this other tool called Percolator to recalculate the scores and end up in a situation like the one on the right, where it is much easier to separate the false and true positives. And so this method was then implemented as a tool called MS2Risker. And by using MS2Risker, we can indeed dramatically increase the number of peptide identifications, even at very low FDR, such as 0.1%, where search engines without rescoring do not find anything. And in fact, the rescoring is working so well that we can set aside altogether the existing search engine and develop a completely new search engine that is based on MS2PIP and DeepLC and is fully machine learning based. And we call this tool IonBot. So the advantage of IonBot is that, again, it has a very low FDR, and because of this very low FDR, it can perform what is called an open PTM search. So instead of specifying a limited number of PTMs to add to the peptides during the generation of the theoretical MSMS spectra, it will just look for any kind of delta mass that corresponds to a known PTM. So the take-home message from this introduction is that when you search PTMs, you inevitably increase your FDR. But if you start from an FDR that is already very low, then you can enable this open PTM search, which then in turn enables the unbiased and comprehensive detection of PTMs, which produce the data that you analyzed. So now I will give you a few examples to try to convince you that visualizing PTMs is very challenging, but also very important. First of all, we usually represent proteins as linear sequences of amino acids, but in reality they possess a 3D structure. And we would expect the modification sites to be on the surface of this 3D structure, because banally, it is the region that is more easily accessible to protein-modifying enzymes. So when we find a modification site that is buried inside the protein, this can mean two things. Either it is a false positive, or more interestingly, it means that the protein undergoes structural changes that expose the site so it can be modified. Similar to this point, there is the fact that PTMs are commonly believed to be localized in disordered region, again, because these are flexible and thus can more be easily bound by enzymes. However, what I noticed when I started to analyze the IonBot results is that only a few PTMs actually are enriched in these intrinsically disordered regions. Most of them are actually found in domains or other more rigid parts of the proteins. So this is something that we will need to look into more carefully, because it kind of defies what is already known in the literature and also a bit of common sense. The next issue with visualizing PTMs is that each site can be modified in different ways. This is especially evident on histone proteins, which not only have a lot of different sites that can be modified, but in addition, each of these sites can bear up to 5, 6, or even more different modifications. And it is important to visualize all of them, because each modification will have a different biological effect. For example, the arginine tree of histone H4 can be asymmetrically or symmetrically dimethylated. The asymmetric dimethylation is a mark of open chromatin, while the symmetric dimethylation is a mark of closed chromatin, so completely different outcomes. And we don't really know how much this is true also for non-histone proteins, because so far there has been a lack of data, but just from an overview of the preliminary results from IonBot, we were able to identify over 10,000 sites with 2 possible PTMs, 3,000 sites with 3 possible PTMs, and another 3,000 sites that have 4 or more possible PTMs. So these sites that are heavily modified are very likely to be biologically relevant. Finally, there is the interaction between PTMs and mutations. Now, in the most common scenario, you have a modified amino acid that is replaced with another that cannot be modified. So you have a loss of the PTM. However, it can also happen that the mutation mimics the PTM or the effect of the PTM. So for example, there is this arginine 256 of nono that when it is mutated to isoleucine or when it is methylated, it can increase the binding of nono to PSPC1, and this increased binding could be linked to colon adenocarcinoma. Of course, mutations not only affect the specific site where they occur, but they can also have a distal effect. So you can have one mutation affecting multiple PTMs, or vice versa, one PTM affected by multiple mutations. And this usually happens because the mutations disrupt motifs that are important for the recognition of the protein by modifying the enzymes. For example, here, if we lose the acidic residue, these kinases can no longer bind the serine or threonine and phosphorylate it. Which, to kind of wrap up the presentation, like if we take the 2 last points together, we, a consequence of these 2 points is that there is also a crosstalk between PTMs, because like the same way that mutations can alter the properties of your amino acid, also PTMs essentially change the physicochemical characteristics of amino acids. So for example, there is this sRGG motif that has been found on some yeast protein. The RGG motif is usually recognized by arginine methyltransferase 1, which methylates the arginine. But when the serine before the arginine is phosphorylated, the negative charge from the phospho group prevents the binding of the arginine methyltransferase. And I said that this is a sort of similar to the effect of mutations, because, for example, to validate this finding, they used a phosphomimetic peptide, and essentially the phosphomimetic peptide just mutates the serine to glutamic or aspartic acid, so that they have a negative charge that essentially has the same effect as the negative charge of the phosphor. And of course, now we are starting to see also some works that try to assess the combined effects of both mutations and PTMs on other PTMs. Like, for example, there is this paper where they study how PTMs and mutations on the N-terminal tail of histones H2A and H4 can affect the N-terminal acetylation of these 2 histones. And so, to conclude this presentation, I would like to thank, of course, my group, all the members of my group, my PI Leonard Martens, the organizers of Bio+MedVis that selected our suggestions for the Bio+MedVis challenge for this year, and of course all of you for the attention. Sorry?
Thank you, Enrico. Do we have any questions for Enrico? I think you can either walk to the stand there or go into Slido and ask questions. In the meantime, I have one question. So all of these 10,000 modifications, for example, do you need to go into and investigate in detail for each of these modifications to do findings, or can you immediately discard a lot of these modifications because you know that they don't matter?
Okay, yeah, that's an interesting point. I would say that for now we mostly don't know. Like there are some modifications that we know they are artifacts. Like banally, there is the carbamidomethylation that you do on cysteines to prevent the formation of disulfide bonds when you do the sample preparation. Methionine oxidation is another common artifact that just occurs during the ionization in the instrument. But yeah, there are also cases where, for example, again, the oxidation of methionine, it can also indicate redox stress inside the protein, because in the end, it's a modification that can naturally occur, and you don't know if it occurred during the acquisition of the spectra, during the sample preparation, or if it was something inside the cell that caused it.
We also have a question from Slido. So it's from Anonymous. Is there a reference database for these various PTMs? Analogous to Phosphosite Plus for phosphorylation changes, for example?
Yeah. So Phosphosite Plus, it's a database. It started as a repository of just phosphorylation, but now it also includes other PTMs like methylation, acetylation, ubiquitinylation. Of course, the phosphorylation database, that dataset is the biggest of the ones that are present in Phosphosite Plus. It of course for each site lists all the reference where the site was detected, but by itself it does not have the quantitative measurements. Then other repositories of PTMs, for now they don't come to my mind. I mostly use Phosphosite Plus. Of course, you can always use UniProt. UniProt is cross-referenced to a bunch of other databases. So if you go to the PTMs and processing section of the UniProt page for your protein, you should find all the most important PTMs on that protein.
Okay, thank you. We could potentially put those links in the Discord channel.
Yes.
Well, let's thank Enrico again. And now it's time for exciting contributions. The first talk will be by Janus Peters. Welcome up. He will be presenting EPROM exploration of protein modifications. Do you mean we can still have this one, but if we were two, or do you want me to switch that one? Remove that one. So, you can switch this off, right?
Good afternoon, everybody. I'm Jannes Peters, PhD student at Hasselt University. And let me start by showing you the proteins we had to work with. So we obtained 3 proteins, each known to be related with a specific rare disease. We obtained both the human and the mouse variant for these. And we had to map the detected modifications to the protein 3-dimensional structures. And so we developed a visual interface that targets researchers in biochemistry, proteomics, and precision medicine to visually explore where protein modifications are most likely to occur and their relationship towards residues with identified pathogenic mutations. This interface contains of 3 major components, being the protein browser, the MolStar viewer, and the detail plot. The protein browser, the protein browser shows the primary structure of the selected protein. So we show the protein sequence horizontally, and the vertical space is used to denote the observed number of modifications on the residues. And note that in here we make a distinction between the actual modifications and the modifications that were classified as artifacts or chemical derivatives. We also annotated the visual with the protein's secondary structure on the main axis and indicated where that the positions where that the mutations were found, and also the ones with active or which are active or binding sites. Now, as the name suggests, this visual also acts as a browser to interact with the other visuals. So we can make a selection on the residues of interest, and note that also all the other residues which are in the selection's 3D proximity are highlighted in gray as well. Next, we have the MolStar viewer. MolStar is a open source tool to visualize molecular data, and we use it to show the secondary and the tertiary structures of the proteins. And so we embedded it in our interface and adjusted it a bit such that we could color the residues based on the number of modifications that were observed. And as you could see, when we make a selection in this protein browser, the MolStar viewer automatically zooms in on that selection that we made. Then we also have this detail plot to provide us more in-depth information about the observed modifications on the set of selected residues that we made and on the residues in their 3D proximity. The selected residues are highlighted with a green line on top of the residue letters, and the residues in their 3D proximity, they have a gray line on top. And we positioned the residues horizontally in order of occurrence on the protein sequence, separating the non-consecutive residues by 3 dots. Modifications are then encoded as circles below the residue letters. And when we hover the mouse on a certain modification, then this tree visualization is triggered, which shows us all the other amino acids that this modification can bind to according to the Unimod database. And based on these, we can then highlight all the other residues in the neighborhood, which this modification could also occur on, representing the measurement uncertainty of the mass spectrometry. Now, let's take a look at this interface in action. Yeah, so we can make a selection from the proteins and then read about the protein's function. We can make a selection of residues that we're interested in. Note that in this particular selection, we found that 2 active and 1 binding sites are located close together in the 3-dimensional space. Also, so when we move the cursor's position is now all times indicated in all 3 visualizations. Clicking on a residue shows us a list of all modifications observed on that residue. And we can color the modifications based on their classification or modification name and use the filter buttons to exclude artifacts and or modifications that are not of interest or classifications that are not of interest. And once again, hovering on this modification shows us this tree visualization showing the other amino acids this modification could have bind to, and the uncertainty of the mass spectrometry using the arcs. So thank you for listening. If you have any questions, feel free to ask.
Thanks a lot. Perhaps Hauke Bartsch can come up here and set up his computer. He's virtual. Then I invite him to come up virtually here and present. Do we have any questions? No? Okay, then let's play the virtual presentation then. So Hauke is presenting mouse-human hybrids. Aaron, if you want to, you could set up your...
And we are presenting a novel way to visualize sequence modifications of proteomics data. People seem to be okay with black box solutions if they can just test them, as, for example, seen in internet searches or with artificial intelligence solutions. How do we apply now those test-based validations to data visualization? In the bottom here, you can see a more traditional user interface where we have one component displaying data and then 3 components that are specific to algorithms with user interface components that are specific to each. We are proposing to replace this traditional style with pattern-driven display. So each of the algorithms is used to derive a number of patterns, and we will map the data the patterns back to the data. This allows us to create a new interface where we are using a simplified control surface to switch between patterns and add the patterns to the raw data. As an example, the proteomics data challenge is modifications of protein sequences of mice and human. The pattern generating analysis, we selected 3 of them. One is marginal distributions, we just show outliers. Frequency analysis, like the slowest repeating frequency in the data, the second slowest repeating frequency. This is treating the proteomic sequence information as a time series. And then a set-based analysis where we are comparing mouse and human and generate displays which highlights what is in the mouse and not in the human, what is the same in the mouse and the human, or, for example, what is added by the human proteomics modification sequence using the mouse. The representation of pattern is very easy. We're using a traditional bar chart display, and the pattern is animated as the height of the bar chart. The control surface using Voronoi tree maps. You can see there the chosen design in the outlined rectangle and on the top. Our control points for that surface are arranged in concentric circles, and on the bottom right you can see the final design for the demo we're about to looking at. In green, the set-based analysis; in orange, the analysis based on marginal distributions; and in blue, the patterns that are derived from the frequency analysis. You can see the user interface has a focus on the raw data at the bottom, and then the control surface on the left. Selecting one of the control surface areas, we can alternate the information between mouse and human. You can see the corresponding part appearing and animated in the display below, or we can use a set where we highlight mouse and human, where mouse and human are the same. If we look at the marginal distributions, the values in the middle, the average values, and their distribution is displayed by a display like this. And if we're looking at the complementary display of outliers, it looks like this. Frequency analysis, the slowest repeating frequency, those are specific to the protein, to the sequence, and they're calculated based on peaks found in the frequency histogram in the spectrum. We are calculating the frequency and phase information, and that's the pattern that is then used to animate. You can see here as you go to increasing detected frequencies, that some of the patterns, that each of the patterns is specific for the proteomic sequence. If there are no detected patterns, then there is no animation indicating that there is no fourth repeating frequency for this component. In conclusion, our user interface is in part designed by an algorithm, which we designed to allow for a scalable visualization where new algorithms can be added easily as intelligent pattern generators. We hope you liked our model interface for test-based data visualization in the field of exploratory data analysis. Please come to our poster and learn more. Hello, my name is Hauke Bartsch. This work is titled Mouse-Human Hybrids, and we are presenting a novel way to visualize sequence modifications of proteomics data.
Have an applause for Hauke. And the next speaker is Aaron Waters. He's presenting Annotated, Taming Protein Beasts Through Interactive Visualization.
This is a brief introduction to Annotated, our submission to the Bio+MedVis Challenge 2022. Annotated is intended to permit visualizations of proteins involved in disease and to allow users to explore sites at which amino acid residues are modified in potentially disease-relevant ways. It is published as a public website as a GitHub repository. The repository includes 2 screenshots, and rather than go through the interface in detail, I thought I'd show you how to reproduce the screenshots. The first screenshot shows the location and type of residues in protein P49312 annotated with O-length glycosylation. Now, if you were coming to the site by yourself, you would click this link here, but I've prepared this so that I don't have to do that. So here's the interface, which is a web application. To reproduce the screenshot, the first thing we do is we go to P49312, wherever that is, on the bottom here. And then you see it's automatically rotating, but I'm going to turn off auto rotation. I don't know why I can't see the rotation. Oh, it took a while to load. Now it's rotating. I'm going to turn off auto rotation and then turn off everything except for O-length glycosylation. And then the little circles indicate where you have O-length glycosylation, and if we rotate the figure, we can see that that all of them are either, I believe, threonine or methionine, and they're all near the surface of the protein, so that potentially could be interesting for an analyst. Now, if we go back to the intro page, we can take a look at the other screenshot. This screenshot is more focused on the disease relative, or disease relevant modifications. So for this one, to reproduce this screenshot, we are going to look at P09. And then we say we only want the pathogenic one. And there's only one pathogenic residue. It's right here. We select that to focus, and then we go back and we look at all of the modifications. And then there's a slider here to adjust the radius. So we can slide down to only look at the residues which are near, the annotated residues which are near the pathogenic annotation. And now we can rotate, rotate to see the geometric structure. And this thing is auto-rotating, so I'm going to turn that off. Okay, turn it off. There we go. All right, the interface is implemented in JavaScript using HTML5 and 2D canvases. It is suitable for inclusion in any web page or web application. And the source data was translated to JavaScript object notation by a Python preprocessor. And thank you very much.
Thank you very much. Should we use that one again, or no? Continue with this one? Okay. So the next speaker is virtual. We have Mo Rahman, who's going to present a simple pipeline for creating cover quality images of post-translational modifications to molecular structures in rare disease.
Images of post-translational modifications to molecular structures that are involved in rare disease. We're going to be creating a simple Unix pipeline to do this. And here you can see the 3 steps that are necessary to do that. The first step will take place completely inside of a terminal that you have available to you, whether in a Mac OS or Ubuntu or any kind of Unix system. And in the second step, we're going to be using our new molecule editor called Dalton. It's cross-platform. You can even use it in the browser. And then in the final step, we will render our complete image, and you can use the renderer of your choice, but we'll be using Procreate specifically here. So let's go to the first step, which is essentially just to understand what grep is. It's a regular expression command, essentially, and it's built into Unix systems, and you can learn more about it by looking at the manual. And essentially how to do it. And what's great about it is it's very quick. It's built into your system. You don't have to code anything new, and you don't have to supply any delimiters. So let's take a look at our challenge data set. And when we look at our challenge data set, we can see that there's several columns all separated by commas. For us, it doesn't matter. Essentially, at the beginning, we see our ascension number, and then which residue, which position, what the modification is, the species, and then finally the last column, whether it's pathogenic or not. So for this example, we're just going to look at TGFB1, but the same applies for all the challenged diseases. So once we type in, grep for TGFB1 human, it'll pull up all of the modifications related to TGFB1. But we don't want all of them. We just want the pathogenic one. This is where the simplicity of the Unix command line comes in. We just pipe it to a second grep command, and this grep command will specifically search for true, because remember, in our data set, every pathogenic modification is labeled as true. And at the end of that, we see we only have about 6 modifications that we need to model and render. If we modeled all of them, it would be a mess. We wouldn't know what we're looking at. 6 we can do and kind of have like a good idea of where on the molecule these modifications fall and sort of how they might affect the function. Once that's ready, we just sort of clean up the output a little bit and cut out only the pieces of data that our molecular editor will need, which is essentially just the residue type, the residue, the position, and anything else that might help us in our rendering and visualization. So now that we have that, we can fire up our molecular editor. Now, before we move on to a lot of the functions of the editor, one thing that we think is really cool is the search ability. You can use natural language to search for pretty much any molecular structure that might be available in a database. So you can just type in something like human coronavirus or rare disease X or rare disease Y, and it'll pull up all the structures that it thinks are related to it. Now, we're trying to make the search better, but it works. Once you have your structure pulled up, it'll just load it right from there. Once you have that, you can now use all of these editing buttons you have to the side. Now, this will allow you to change colors, remove atoms, add atoms, what have you, and essentially get your structure ready for the final rendering. Now, the final rendering we're going to be doing in Procreate, which is a tool for iPad, and you can just import your 3D structure into that after you've edited it in the molecular editor. And it has this great function called 3D Paint, which will allow you to paint the structures the way you want, with the colors you want, and add typography, everything like that. And the final result will be a beautiful rendered molecular structure with your post-translational modifications that we think would look beautiful on the cover of any journal. Thanks.
The next speaker is Nicolas F. Chavas de Plaza. He will present Protofold Neighborhood Inspector. It's also a virtual presentation.
At TU Delft. Today, I will show you the Protofold Neighborhood Inspector. Post-translational modifications affecting a protein's residues can disturb its function, leading to illness. Whether a PTM is pathogenic or not could depend not only on its type, but also on the status of neighboring residues. The Protofold Neighborhood Inspector is a visualization system for analyzing residues neighborhoods. The visualization system supports 3 tasks: selecting residues for neighborhood analysis based on a residue and neighborhood level characteristics, identifying patterns in the distribution of a neighborhood's features, and comparing neighborhoods to assess their similarity or dissimilarity under a feature of interest. 3 datasets support the visualization system. The structure dataset provides the spatial coordinates of the protein's residues as predicted by AlphaFold. The modifications dataset lists pathogenic and non-pathogenic PTMs that affect the residues in the structures dataset. We derived a third dataset, neighbors, which for each residue lists its 30 nearest spatial neighbors. We use the Euclidean distance as a metric to find the neighbors. We now proceed to present the Protofold Neighborhood Inspector. We start by explaining the novel residue constellation idiom and then describe the supporting widgets. The residue constellation idiom permits users to select residues, analyze neighborhoods, and compare them using 3 components. The first component is the primary structure orbits, which allow users to select residues for neighborhood analysis based on categorical and numerical variables. There are 3 orbits. The first one depicts the residue position in the protein's primary structure using a sequential color scale. The second orbit depicts a user-selected categorical variable. Examples of categorical variables include residue type, pathogenic status, and a structure type. The third orbit encodes the value of a user-selected numerical variable using the height of the arc sections. Users can leverage the orbits to select residues that call their attention. Examples are residues that share a variable value, have a given prediction quality score, are located in a specific part of the protein structure, or have certain neighborhood characteristics. The second component of the residue constellation is the neighborhood summarization glyph, which permits users to analyze neighborhoods of residues selected with the primary structure orbits. The bigger circle corresponds to the selected residue. Surrounding circles correspond to unselected neighbors. The stroke of the outer circle shows the pathogenic status of the current residue, The stroke width of the inner circle encodes the prediction quality, allowing users to analyze the data under uncertainty. The fill of the inner circle summarizes the pathogenic status of residues in the neighborhoods. For instance, if a neighbor is pathogenic, then the inner circle will have an orange color. Finally, the PTM pie shows the distributions of different modifications in the selected neighborhood. The stroke color of the different sections of the pie encodes whether the modification was pathogenic or not. During the analysis, the user can change the neighborhood size, updating the summarization glyph accordingly. If a neighboring residue that contains a pathogenic modification appears, the color of the inner circle changes to reflect the change of neighborhood's pathogenic status. The final component is the neighborhood force layout, which leverages summarization glyphs to permit the user to compare different neighborhoods in the protein. In addition to the residue constellation, the Protofol Neighborhood Inspector provides several widgets to support users' analysis workflows. A transient ribbon chart allows the user to obtain a preview of a residue's neighborhood. A 3D structure view permits users to understand where the selected residues are in 3D space. Finally, a bar chart of the frequencies of the current categorical variable lets the user select residues that share a given value of the variable. To conclude, the Protofol Neighborhood Inspector permits users to select, inspect, and compare residues neighborhoods through the novel residue constellation idiom and supporting widgets. For future steps, we plan to implement filters based on combination of multiple variables, derive neighborhood level metrics like pairwise similarities, and extend the residue constellation idiom to handle multiple protein structures. Thank you for your attention.
Yeah, that was the contributions for the main challenge, excellent contributions. Now it's time for the redesign challenge. And then I first invite Enrico up here again to present this, or to introduce the second part of the challenge.
Yeah. So the second part of the challenge was a sort of a simplified version of the main challenge, where we provided a starting point, which is the focus was the 2D representation of the protein sequence with all the different PTMs. And so the focus of this secondary challenge was mostly how much information can we fit in a 2D representation of your proteins without making it too cluttered and essentially unreadable. Like, for example, here it is already pretty hard to tell which are the residue of interest, and like this is already the zoomed version of the image. If you look at the image that was on the challenge page, it's even more unreadable. So yeah, this was the redesign challenge.
Oh, thanks. Then the first speaker here is Hyun Nguyen. She will present ModiViewer protein beasts and how to view them.
Okay, hello everyone. Thank you for having me. My name is Huyền and I'm from Texas Tech University. I am a PhD candidate in computer science. So today I will be here to present our work with my co-authors, Caleb Trujillo and Tommy Dang. Our work is multi-viewer, protein bits and how to view them. The bits here, of course, are the protein modifications. So we are provided with an existing visualization. The issue with this visualization arises when we wanted to have a comprehensive view of the whole protein sequence. So the first issue is that overplotting with the horizontal space. The second issue is about the vertical order of all of these circles representing the modifications. So what are these order stands for? And the last issue is the color encoding is very ambiguous. What are the information that are encoded in these color? So to address all of these challenges, we have built a comprehensive application with 3 views. First of all is a classification on protein modifications. The second view is for modification distribution. And the third view is for modification type. We also tackle the 3D structural problem. And we have an online platform, and there's a link and QR code in the corner if you want to try it out. So for today, in this talk, I will focus on the classification of protein modifications. And for the solution, we come up with 3 main strategies. The first thing is horizontal stacking. What is that? So on the horizontal axis, we present the modifications. On the vertical axis, we present the classification. The stacking happens when there are more than one modification belonging to the same classification. These modifications will be stacked horizontally, and these modifications are color encoded by the classification. These classifications are, for example, artifacts, glycal, or multiple. And then the third strategy is ordering algorithm by similarity. That is when we need to order all of these around like 10, 11 classification on the vertical axis. And we group them by similarity so that we can observe the patterns of these similar groups of classification. So in here, we also incorporate user interaction. So in the first panel is the overview of the whole protein sequence. And we use focus and context incorporate with brushing and linking to zoom in the middle part of the upper panel. Here, the mutations are marked with X. So using focus and context, we can see that all of these mutations are, when they are belonging to the modifications that are classified, they belong to either post-translational, chemical derivative, or artifact. So our platform is available online, and the multi-viewer is built with horizontal stacking, focus and context, and ordering algorithm. So thank you for listening, and that is the end of my talk. Thank you.
Thank you very much, Yuan. The next speaker is Daniel Mbott. He will be presenting EEPROM, Exploration of Protein Modifications.
Okay, thank you. I'll just dive in straight to our submission because we've already had a nice introduction to the redesign challenge. And our submission came from the discussions and the sketches that we made for the main challenge that Jonas presented earlier. And we ended up with a radial design where we show the modification names along the circle and the residue positions along the radius. Here, the beginning of the protein sequence is in the center and the ending is at the outside. The actual modifications are shown as circles in this space, and we've colored them using a color scheme that captures the category in the classification. So, for instance, all the translational classes are a shade of blue. The artifact classes, we gave them a very neutral color, and we put them on the background so that they're still there, but they don't really capture the attention. We've added a red circle and a line that captures the mouse position, just to improve readability a bit, because it can be quite tricky sometimes. We've also added a sidebar that has several filtering options, and we've added a vertical version of our protein browser that shows the secondary structure, and you can use that to zoom in on specific regions if those are particularly interesting. So in terms of visual encoding, this design is doing okay. You can argue that it's using position for the two most important attributes of the data. You could also say it's using angle for the modification name, which is not quite as good for an ordinal data category, but yeah, it is okay. In terms of color, we use hue and saturation for the nominal and ordinal aspects of the classification, so that's also a top 3 option. So that seemed okay. However, we used a radial design instead of a Cartesian one, and you can say that's a bit of a controversial choice. The reasons we chose this design were mainly aesthetics. We felt this was more of a signature than the Cartesian designs we had. And there's a bit more space for the labels on the outside. However, half of them are upside down, so they're not really all that readable. And there's an issue with space allocation, particularly in the center, because there's still some clutter going on there. In terms of the radial design, literature doesn't seem conclusive yet, but they can be more difficult to read, and there is no inherent circular aspect to this data. So yeah, you can argue whether that was a good choice. And I've prepared this slide just to show what that looks like visually if you add a Cartesian version. That's on the right here, and the original version is on the left. And but the benefit of a Cartesian version here is that all the modifications of a signal residue lie on a line, which is easier to follow than a full circle. However, it needs more space to show all the labels. And actually, if we had done an evaluation of our design, we might have ended up with the middle one, where we flipped the axes. So now it's the positions around the circle and the modifications along the radius, so that the center doesn't have overplotting because we put the least occurring modifications there, which would have been interesting. But that's all I have for you today. And if you want to try our designs, you can scan the QR code and go to our website.
Thank you very much. Our next speaker is Laura Garrison. She will be presenting visualizing protein residue chemical modifications.
All right, so this was our stab at doing a redesign. So this was with myself and colleagues Hauke Bartsch and Stefan Buchner from the University of Bergen in Norway and the Moon Medical Imaging and Visualization Center. So this has been briefly discussed, but I'll just cover it quickly. So the original visualization shown here is not super ideal for the task because the visualization is very cluttered, so it's difficult to identify modifications occurring at certain sites. And when you have multiple modifications, it's so overplotted, it's difficult to understand what exactly is going on. So our redesigned visualization, which is shown here, consists of 3 linked views to facilitate identification. Oh, closer. Oh, okay, sorry. Is this better? Okay. Yes, so we have 3 different views. We use a focus context approach with interactions that you can see here to enable a comparison of modifications per protein site within and between species. We retain the sequential structure of residues from the existing visualization. This echoes common practice for visualizing omics sequence data, which is familiar to domain experts. Positioning protein residues in this linear ordered sequence enables users to quickly identify sites or single sites on which different modifications have occurred. We exclude unmodified residue sites, and this is similar to the practice of visually filtering out introns in genomic sequence visualizations, and this removes uninteresting regions from analysis and economizes screen space at the same time. We describe modification frequency on 3 levels. At the coarsest level, which is shown in the topmost panel, we summarize per site modification diversity, which is the number of modification types and their proportion in a given protein sequence. We do this with Shannon's diversity index, calculated as the negative sum of p sub I, where p sub I is the proportion of a modification type at a given site, times the natural log of p sub I. And the index iterates over each type of modification present in that site. A high diversity index means that the site has relatively more different types of modifications present, and that each type of modification occurs in roughly similar amounts. Conversely, a low diversity index indicates a low diversity site, for example, with only one type of modification or that's present in low amounts. And we map this value to luminance, and we like this because it looks sort of like a PCR gel. And residues can be sorted in ascending or descending order of their diversity index. The next level shows modification position and frequency of each modification type for each site, and this is shown in the middle panel, which uses, like a colleague earlier, a stacked bar metaphor. Stacking the bars captures the overall modification frequency on a site, as well as the proportion of each type within a site. And subset options that are available in the right panel permit exploration of the per site frequency of a single modification type. And this can help solve the perceptual issues that can arise when you're trying to estimate relative frequencies when they're not on the same baseline. Each modification type is mapped to hue in an unordered colorblind accessible color map, which is shown in the right panel again. And just like in the other view, you can sort according to mutation frequency in ascending or descending order. Our most fine-grained view focuses on the modification types and their frequencies for a single site, which has been selected from either of the 2 coarser grain context views. We're using a unit visualization metaphor to emphasize the individual countable occurrences of modifications within each site. And this encoding is inspired by prior work on unit and icon visualization, such as by Park and others or Rose and others. And we use a simple square glyph to encode each occurrence of a modification, and this reduces visual complexity and retains coherence with the other stacked bar chart with a similar color map. So we mock this up in Tableau. If you want to check it out, you can go to this QR code, and we also have a poster available, so if you want to come hang out and chat, I'll be happy to chat with you then. Thank you.
Thank you very much, Laura. It's time for our last contribution by Simon Hackel, a dashboard for the visualization of proteins, PTMs. And it's a virtual presentation.
Atomics group at the University of Tübingen, and I'm going to talk about our group submission to this year's Bio and MedVis Redesign Challenge, a dashboard for the visualization of a protein's PTMs. So what you see on this slide is the starting point of this year's redesign challenge. The main points we wanted to address with our redesign proposal for the visualization of all PTMs of a protein were firstly to eliminate the clutter of graphical elements, that is to say, to provide more space for the diversity of PTMs, and secondly, adding information from the protein structure. So we wanted to associate the PTMs not only with the primary sequence of the protein, but also with the 3-dimensional structure. We have developed an interactive dashboard to visualize the PTMs of a protein, composed of different components, which I will talk about in the next few minutes. The first component you see here is a PTM presence-absence map. Here, each row represents a position in the primary sequence of the protein, and each column represents a PTM type. Filled cells indicate that the PTM was observed at a given protein position. With this component, the PTMs of a protein can be explored without the visual clutter being a hindrance. In addition, specific PTM patterns along the protein sequence or PTM variability at a given position can be compared. The second component is a two-dimensional contact map-like visualization in which the primary sequence positions of the proteins are plotted against each other. Here, a filled cell indicates that the two corresponding protein positions have a distance in three-dimensional space below a certain fixed threshold. In general, this reduces the complexity of 3D protein structure visualizations by projecting local neighborhoods onto a 2D visualization. While we have left the lower left triangle of this visualization unmodified, the upper right triangle has been altered to include information on the PTMs. When both residues are in contact and modified, cells are either colored gray if no shared PTMs are present, or colored according to the ratio of shared PTMs. Cells that have residues in contact with at least one unmodified residue are not colored. This visualization relates the observed PTMs to the local neighborhood of the residues and has the potential to reveal clustering effects of PTMs on protein structure. Our dashboard now consists of these two main components, the PTM presence-absence map on the right and the contact map-like visualization on the left, both with a shared y-axis. In addition, we complemented the contact map-like visualization with single-axis traces that display information about the secondary structure per position. For example, whether being in a helix, a beta sheet, or loop. Furthermore, we added bar charts at the top and right of the dashboard showing the total number of PTMs per primary sequence position to summarize the number of modifications per position. Finally, let me show you what our dashboard looks like with real data. Here with the dataset of the human aldolase A protein, one dataset provided for the visualization challenge. We choose a C-alpha atomic distance of 6 angstroms as the distance threshold for the contact map-like visualization, and use the Shakar index to calculate common PTMs for neighboring residues. For the presence-absence map, the secondary structures are projected as the background color of the rows, and the PTMs are sorted alphanumerically. In the top stacked bar chart, the number of PTMs is distinguished into those that are common to neighboring residues and those that are unique. The dashboard is implemented as an interactive web interface that allows users to zoom into regions of their interest and pan around. Thank you for your attention.
Thank you very much for attending the first part of the challenge. We will take a 30-minute break and then we'll be back to hear the invited talks. See you.